Lastly, once the light is inside the skin, it is drawn to the pigments present in that area. The attraction transforms the beam energy into heat. This phenomenon is called absorption. Therefore, when we expose a white surface to the sun, it does not heat up since white reflects the entire light. The opposite occurs in the case of a black surface, because it will absorb all the light. The pigments in the skin are called chromophores. The first one is called melanin and is responsible for the color of skin and hair. The second one is the hemoglobin, the pigment in the red blood cells. And the third one is the water. When we look at the rate of absorption of chromophores, we see that melanin is absorbed particularly well between 300 and 400 nanometers. This explains why it plays such an important role in protecting skin against the ultraviolet rays of the sun. Therefore, skin that has little or no melanin is poorly protected against the harmful effects of the sun. Hemoglobin is absorbed in a series of crests, or at high points, the best of which are found at 400, 540 and 570 nanometers. Water absorbs the infrared beam best from 1000 nanometers. Due to the fact that cells are composed of more than 60% water, if the number of infrared rays is too high, heating will become too intense and create the risk of cell destruction. It seems therefore that there are certain targets in the skin that draw light. This is why in many cases the target is an indirect target. And if the target does not contain pigment, the chromophore or colored portion of a neighboring object becomes a target instead. Heat generated by light hitting a chromophore is transferred simply by being in contact with the neighboring target. The rate of heat transfer is very important. This notion is called thermal relaxation time, or tissue cooling time. It varies according to the type of tissue and the duration of the pulse, as well as the amount of energy in the pulse. We can compare heat transfer to what happens when we try to fill a container that has a hole in it. If we fill the container more slowly than the rate at which the liquid is emptied, the container will not fill up. If, however, the rate at which the liquid enters the container is greater than the rate at which it leaves, the container will only fill to a certain level. This level corresponds to a balance that will not change as long as the filling and emptying rates remain constant. The same thing applies to a target and heat. If the cooling rate is slower than the heating rate, the heat will remain concentrated in the target and will be efficient. On the other hand, if cooling occurs more quickly than heating, the target will not be able to remain hot, and nothing or very little will occur. This is why for best results it is important to vary the pulse width as well as to regulate the power levels so the different targets will all be treated correctly.